So at this point in the process, we have talked about transcription, which is copying a gene, which is located on the chromosome in the form of DNA, copying that into a single-stranded RNA. And then for the mRNA type of RNA, we talked about some modifications that are made before that RNA comes out of the nucleus, because keep in mind, the DNA is in the nucleus. The mRNA travels out of the nucleus to the ribosome, and the ribosome will translate the mRNA, the codons, and build a protein, and the protein will fold. And so where we are now in the process is the proteins are targeted or trafficked to their final destination. So there has to be a system to move the proteins to where they need to go. Some proteins will just stay in the cytosol, but you may recall there are enzymes for cellular respiration that are in the mitochondrial matrix. There are enzymes in the lysosome. There are different proteins that are found everywhere, proteins that make up the cytoskeleton. So proteins, whatever their function is, there's also a location typically where they must be delivered. And so when we talk about protein targeting or trafficking, it means what you think it means, I think. It's that the protein has to be moved to where it needs to go. Because if the protein is correctly formed and folded, but it's not in the right location, it can't do the job that it's meant to do. And so typically the way this works is in eukaryotes, all translation begins on what we call free ribosomes, ribosomes that are just in the cytosol. And then what happens is, remember, those two subunits of the ribosome come together and sort of sandwich that mRNA and start sliding along till they find the start codon. And then the methionine is added, and then the next amino acid, and the next one, and the next one. And so what happens is as that protein is being built, it starts to extend out of the ribosome. So the first part of the protein is poking out of the ribosome while the rest of it is still being made. And that first part, if that protein needs to be targeted or trafficked to a different location, the amino acids that are in that very first section are what we call the signal, or the signal sequence that directs another process that will move that, not only that protein, but the ribosome that's still building that protein to the correct location. And so in this one, I'm going to describe for proteins that need to go maybe out of the cell or to the Golgi. What happens is the signal will cause those kinds of proteins with the ribosome and the mRNA still all connected. There's a particle called the signal recognition particle that latches on and moves the whole thing over to the outer surface or the cytoplasmic side of the rough ER. And so the ribosome docks on a little channel at the rough ER, and the protein is threaded through that pore, and the protein, when it's finished being synthesized, will be released into the lumen of the rough ER, and then it will fold. And then the ribosome doesn't stay attached to the rough ER. It will then detach and just become a free ribosome again. So ribosomes are free when they're not attached to anything, or they're bound when they're attached to the rough ER. But it's not a permanent situation. Ribosomes that are free can become attached if they are translating a protein that needs to be put into the rough ER lumen, and then when they're done with that, they detach. So it's a dynamic process. The ribosomes don't stay in one place necessarily. They're moving back and forth. But in any case, let's talk about what happens to the protein. The protein is deposited into the rough ER lumen, and that's the beginning of protein trafficking. And we've talked about this process before. This is where the protein goes by vesicle then from the rough ER to the Golgi by vesicle. And in the Golgi, it gets modified and sorted and then sent by another vesicle out to its final destination, maybe the plasma membrane for secretion, or maybe some other part of the cell. So here's a picture of what I was describing, and this is kind of a time lapse. So on this far left side here, this would be like at the very beginning, 1. Here's the free ribosome. It started to translate the protein, and you can see the protein sticking out right here. That's the first part of the protein. The methionine would be the very first amino acid right here. And then this section is called the signal. And so the signal recognition particle here will attach to it. And then you get this. So here's the signal recognition particle where I wrote the number 2 attached to the signal and attached to really the whole ribosome and mRNA. And it causes the whole thing to move or dock at a place on the rough ER lumen. So this is the rough ER membrane. So this is the rough ER membrane, and here is this docking site, and here's a little channel right here, and you can see it's threading that protein through that protein channel. And so this is number 3 here. And then the same thing a little bit later, you can see that the protein is now being pushed into the lumen, and then ultimately it's released like this. So that would be, I guess, 5. And at stage 5, it's not really an exact number, but the one I wrote as 5, the ribosome will have detached from the rough ER membrane at that point. But then what happens to this protein is that it goes by vesicle to the Golgi, and then it goes to whatever its final destination is. So what I'm showing you here in terms of protein trafficking is what I would call the prequel story to the protein secretion story that I told you much earlier in the course. So this is how that protein is made and delivered to that rough ER lumen. All right, the last section of this chapter is what happens if the gene, the DNA sequence, has a mistake, has a mutation in it. And there's a couple types of mutations that are common in DNA and genes. One is called a base substitution, and that's usually where you had probably a mistake during DNA replication, or there can be chemicals that cause changes in the nucleotides. But for whatever reason, if there's a nucleotide, let's say a G, at a very particular position that it should have been an A, then that would be called a base substitution. So it's where one nucleotide is in the gene, but it should be a different nucleotide. So the incorrect nucleotide is the mutation. And there can be different effects of a base substitution mutation. If it's, like I said, if it's occurring in a gene, then it could affect a codon. In other words, the nucleotide that should have been there is not there. A different nucleotide is there, so the codon that should have been is changed. And so, based on that, not only do we name it a base substitution mutation, but we can also name it according to what the effect is of that base substitution on the final polypeptide after it's been produced, translated. And so if the base substitution doesn't change the codon to a different amino acid, because you'll remember that some of the amino acids have more than one codon that can indicate the same amino acid, then that's called a silent mutation. So no effect. There's a change in the DNA, but there's no effect in the protein that's produced from that. A missense mutation is where one codon gets changed to a different amino acid codon. So you would get one different amino acid in that protein after it's produced. And then a nonsense mutation, this one is typically considered the worst kind. is where an amino acid codon gets changed, and the change causes it to become a stop codon. So here's some examples, and they're not showing the whole, they're just showing a really short example of a, so you start with methionine here. So here's the DNA is up here. Let me get my pen. And then the DNA is copied into RNA. So here's the DNA here. This is the double-stranded DNA here. That's supposed to represent the gene. And then this is the mRNA that was copied during transcription. And if you have, this is methionine. All right, this is the normal, by the way, they're showing you the normal. In the normal sequence, you start with methionine, then the next codon is for proline, then the next one's for tyrosine, then arginine, and then a stop codon. This is literally a very miniature example. Real genes have thousands of codons in them, but just for this purpose, we're doing a very short example. So let's say that's the normal sequence. And then let's say in part B, let's say you had a mutation, look here. And in this one, at this highlighted area, there's a change. It was a T and an A, but now it's a C and a G. So you can see here, the C and a G has taken the place where in the normal it was a T and an A. Okay. So what that will do, if you look at it, if you look at the genetic code, you'll still get a methionine, then a proline, then a tyrosine, and then an arginine. So there's no change in the protein. There is a change in the DNA, which is copied into the mRNA, but it doesn't change anything. So we call that silent. Silent mutations, nobody pays any attention to them. All right. Now, the next kind, part C, here shows you what's called a missense mutation. A missense mutation is shown here. So this AT was supposed to be a GC over in the normal sequence. So there's a change to an AT that gets copied into the mRNA, and you can see here that creates a new codon. Histidine is placed here at the 4th amino acid position, but it should have been arginine if things had been normal. So missense means one amino acid is changed. It depends on what amino acid is changed. Some amino acids are more critical than others for the function of a protein. So anytime you have a mutation, each case has to be studied individually to figure out the seriousness of the effect on that protein. And then the last one is the nonsense mutation. Again, using the same example, but here you have an AT that happens right in this position, and it should have been a T and an A, so it's switched. And what that does is it creates a stop codon where there wasn't meant to be a stop codon. So UAA is a stop codon. And there's not supposed to be a stop codon there. So that change creates a protein that's shorter than it should be. And that's called nonsense. All of these are all base substitutions at the DNA level, just single base changes, single nucleotide changes at the DNA level, but they're different. We name them differently based on their effect on the protein level. The other main group of mutation you have is when you have an insertion, or what you could call addition, or a deletion of one nucleotide. So in this case, it's not a swap out of one nucleotide for a different one. There's actually an extra one that's accidentally added in somehow for some reason, or one that's deleted out. And just like I said before, sometimes this happens naturally during DNA replication, but sometimes this happens as a result of chemicals and other mutagens in the environment. But for whatever reason it happens, the effect that it has on the protein, it always affects the protein. Frameshift mutations are always affect the protein. There's no such thing as a silent frameshift mutation. And so what typically happens with a frameshift mutation is one nucleotide is deleted or added, it resets how the codons are going to be read at the ribosome. And so everything is now one nucleotide off of what it should have been. So all the codons after that insertion or deletion are changed. And there's typically going to be a stop codon that shouldn't be there, but suddenly comes into frame. So frameshift mutations are always detrimental, always harmful, always change the protein. So those are like single nucleotide, small mutations. You can also have entire chromosome mutations. And in this context, when we talk about a deletion or duplication, we're talking about a much larger region of DNA that's missing, not just one nucleotide, but like a whole section. It could be on a chromosome, it's probably thousands or millions of nucleotides that are deleted from a chromosome. So a deletion on a chromosome is where a large piece of the chromosome is missing. And just like before, there's different reasons why this could happen. But as you can imagine, the effect of a deletion of a very large section of DNA from a chromosome is going to be even more severe than just a deletion of one nucleotide from one gene. A duplication refers to having an extra piece. Usually a tandem duplication means the same sequence is, for whatever reason, repeated in a chromosome, so that chromosome is longer than it should be. You can also have an inversion. An inversion is where a chromosome, for some reason, breaks in two places, and the piece in between those breaks flip-flops and rejoins. And so in this example, you can see how B, C, and D has sort of done a flip-flop and then a rejoin. And then a translocation is where two non-homologous chromosomes exchange non-equal, non-corresponding segments. And so in this example, you can see how KLM ends up on this top chromosome and ABC ends up on this bottom chromosome. And these are not homologous chromosomes. So it's probably the result of a crossover that never should have been. So it's a messed up crossover. Probably an inversion is a mistaken crossover. And duplications and deletions, there are reasons why these probably happen, but we're not worrying about why they happen. We're just describing what they are. So a deletion is where a chromosome is notably shorter. A chromosome duplication is where a chromosome is much longer. In inversion, you can't tell very easily with an inversion, but experts can use certain stains to figure out if there's an inversion on a chromosome, because the chromosome itself would stay the same length, but a piece in the middle has flip-flopped. And then reciprocal translocations, those can happen in pretty much any way. So sometimes it ends up with a longer or shorter chromosome, sometimes not. But these all create these very, unusual chromosomes, abnormal chromosomes, and they create all kinds of problems for the organism. So mutations, whether they are single nucleotide mutations or whether they are these large chromosomal mutations, these are the starting point. These are important for evolution. In evolution, organisms have to be able to have some genetic change, but there's a balance. Too much change is definitely harmful to the individual. And you will have some changes that are harmful to individuals and they don't survive very well. But if you have a low level of change, what will happen is you can get some mutations, and occasionally some of those new variations, those new mutations could actually be helpful to the organism and be favored in natural selection. But the concept of mutation is that there has to be a balance between how much new variation happens and how healthy the species is in its environment. So too much mutation is bad for the organisms. Too little mutation is going to hinder evolution of the species. So there has to be a balance there, and there is. Okay, so just to review the main topics of this chapter, transcription and translation are the two processes that make gene expression, or the central dogma of biology. Transcription is how cells make RNA. I've listed some of the details there. Translation is how cells make protein. And we've gone over those. I would review back over those videos, and I will, they're in the PowerPoint, so you can open the PowerPoint and just watch the videos themselves. And that covers our very last chapter, chapter 15.